Hello and welcome to my next video on animal responses. To start off with, I've got a picture of the brain and I've underlined the 4 key areas you need to know. That is the cerebrum, the hypothalamus, the medulla oblongata, just the medulla here, and the cerebellum. Now we're going to go through what each of these parts do. To start off with the cerebrum, it's split into 2 hemispheres, and the left side controls the right side of the body and the right side controls the left side of the body, and it's connected by the corpus callosum, as you can see in the picture. This region is associated with higher brain function, so that's conscious thought and emotional responses, the ability to override some reflexes, like if you try hard enough you can stop yourself blinking for a certain period of time, and features associated with intelligence such as reasoning and judgment. Now, in the cerebral cortex, which you can see labelled here, so that's just part of the cerebrum basically, there are different areas. There are sensory areas which receive impulses indirectly from the receptors. There are association areas which compare inputs with previous experiences in order to interpret what the input means and judge an appropriate response. So basically, let's say you have before come into contact with an icicle and it has cut your hand. It means that your brain might go, well, we're not going to touch that icicle again because it's associated that. And motor areas send impulses to the effectors. Those are muscles and glands. Now, the next area we're going to look at is the cerebellum, and that's coordinated motor responses. So, conscious decisions are in the cerebral hemisphere, or cerebral cortex. That's not going to actually make things happen. You need to have the cerebellum to actually coordinate it all and also do some non-conscious operation. So let's say in your cerebrum you go, I want to pick up something. I'm consciously going to pick up a glass of water. Well, that's not going to actually make anything happen. What's going to happen is it's going to go to the cerebellum and then it will send out information to make the actual thing get picked up. So, for example, you want to pick up a glass of water, so your cerebrum will go, yes, I want to pick that up. The cerebellum will then be notified of this and go, right, we need to get this moved, and will send an impulse to the motor areas, which will then move your muscles in whichever way is needed to pick up water. Now, some other things that the cerebellum does, it's involved in muscular activities, which respond to changes in body position and remain balanced and upright so you don't fall over. Sensory activities such as judging the position of objects and limbs. That's why, for example, if you close your eyes, you can still touch your nose. You know where your nose is, even though you can't see it. The tensioning of muscles in order to manipulate tools, instruments effectively. So that's going to be like when you're picking up a glass of water. Feedback information on muscle position, tension and fine movements. So, for example, the body needs to be able to know where your muscles are at all times, because if your body doesn't know, let's say your arm is relaxed and not clenched and tries to pick up water, it's just going to knock it over, it won't be able to pick it up. And operation of antagonistic muscles to coordinate contraction and relaxation. We'll look later at muscles, but a lot of muscles in the body are antagonistic, and if you think of the best example is the arm, you have biceps and triceps, and basically it's one contracts, the other relaxes. So when you contract your biceps, your triceps relax. When you relax your biceps, your triceps relax. Not collapse, that'd be terrible. Now, the cerebellum actually contains over half of all the nerve cells in the brain, and it plays a key role in coordinating balance and fine movement, as we said, and it does this using sensory information from the retina. So if you are looking at something, you know dead on and you're not rocking about, you're probably quite well balanced. The balance organs in the inner ear, you have a liquid in your ear which can move and judge how balanced you are. That's why if you're in the car, for example, as we just said, your cerebellum takes information from your eyes and your ears. Now in a car, if you're looking, you're actually relatively stable according to your eyes, because you are moving with the car. So you're not actually, according to where you are in the car, you're pretty stable. So if you're looking at, let's say the dashboard, you look at that, you're not moving in a different way to how the dashboard's moving. So the cerebellum is basically going, that means you're pretty well balanced there. according to your eyes, but your ears are rocking about the place because the whole car as a whole is moving, which means that your ears are going, you're not balanced, you're rocking, this doesn't quite make sense. So your cerebellum is going, right, the eyes are saying you're balanced, your ears aren't, what are we going to do? And you start feeling ill. That's why if you ever feel car sick, it's always best to look at somewhere outside the car directly in front of you, because it means that you are looking at something that's moving in the same way the car is, so both of them will make sense. Other things are specialised fibres in muscle cells called spindle fibres, which give information about muscle tension and the joints. And the other two we have looked at in Unit 4. The hypothalamus is basically the homeostatic centre, and the medulla oblongata is the kind of regulates the cardiac centre, so heart rate, and also has the respiratory centre, control breathing and regulate rate and depth of breathing. But that's just going over Unit 4 stuff. Now, the nervous system. We have looked at the nervous system before and how it actually works, but we now need to look how it is organised. And it's basically, you have the CNS, which is the brain and the spinal cords. Everything else is the PNS, the periphery nervous system. So that's all connected to the central nervous system, but it is its own nervous system. That is split into somatic and autonomic nervous system. Somatic is impulses from the CNS to skeletal muscles, which are under voluntary control, and the autonomic neurons carry impulses from the CNS to cardiac muscle and to smooth muscle, which are not under voluntary control. So basically, somatic is voluntary, autonomic is involuntary. And then in the autonomic one, you have the parasympathetic, which generally lowers things, and the sympathetic, which generally raises things. So the sympathetic will increase heart rate. I'm trying to think of other things. It will cause pupils to dilate more, increase ventilation rate, while the parasympathetic will lower heart rate, constrict the pupils, which means you get less light coming in, so you're in probably a lighter area, and decrease ventilation rate. So basically, parasympathetic is when you're at rest, and sympathetic is when you are stressed. Now I'm going to just compare these two quickly, and we'll look at some other stuff in a minute. But firstly, the parasympathetic. Now you need to think of these things called ganglions. These are basically groups of nerve cells, little lumps of them, and they act as relay points between neurons. Now in the parasympathetic nervous system, you have preganglionic neurons are very long in length because the ganglions are found within the target tissue. So these relay points are found within the target tissue. So you think about it. Think of a neuron going from the central nervous system to the target tissue, and then it has an effect. Now the preganglionic one will go from let's say the brain all the way to the target tissue, let's say the heart, and then once in the heart it will reach a ganglion, and then the post-ganglionic neuron is very short, just going directly to where it's needed in the heart. In the sympathetic nervous system, a ganglion is found just outside the spinal cord. So let's say in this case it's going to the same pathway, so it goes from the brain down the spinal cord to a ganglion. That's a very short distance, and then it goes from the ganglion to the target tissue. That's so it's very long. So just to clarify, parasympathetic, long preganglionic neuron, short postganglionic neuron. In sympathetic, it is a short preganglionic and a long postganglionic. Basically, the best thing to do is actually learn one of them and then know that the other one's the opposite. Another example. In the parasympathetic, postganglionic neurons secrete acetylcholine as a neurotransmitter. And in the sympathetic one, they secrete noradrenaline. Again, you should remember that from Unit 4. So the sympathetic is going to secrete noradrenaline, parasympathetic secretes acetylcholine. And we've already discussed the different effects they have. Now going back to the central nervous system, it is also important to mention that they contain grey matter, which is non-myelinated nerve cells, and white matter, which are longer myelinated nerve cells. Myelin basically makes the fibres appear white, so white matter is myelinated. Then to the autonomic. Now remember, autonomic sounds a bit like automatic, so it's the kind of involuntary one, it naturally happens. Now they're generally non-myelinated. Most somatic ones are myelinated. Autonomic connections to infectors always consist of at least 2 neurons. Somatic connections consist of only one. And then, as we said, the autonomic is split into parasympathetic and sympathetic. Now, muscles. This is probably one of the harder topics in Unit 5, especially when we get to the sliding filament model. But, right, 3 types of muscle you need to know about. Smooth, which is involuntary muscle, cardiac muscle, and skeletal muscle, which is voluntary muscle. So, I'm going to start off with smooth muscle. Some key things you need to know: it's uninucleated, it's got one nucleus. It is slow to contract but equally tires slowly. And some examples are the walls of the gut, the iris of the eye, and the walls of the arteries, and arterioles, walls of the uterus, all those various examples. Now, an example I've drawn here is the walls of the intestines. And confusingly, I've drawn circular as lines and longitudinal circles, but don't worry about that. Right, the centre of it is the lumen. So that is where all your food's going to be passing through. Now, why do we have 2 lots of muscles, circular and longitudinal? Well, circular muscle causes contraction, causes segmentation. Basically means it goes in and out. So that circle's going to get bigger and smaller. But then the longitudinal layer makes it go in wave-like contractions. So the circular makes it go in and out, but longitudinal makes it go along. So you get a contraction in and it pushes it along. So that way you're getting food passing through. And there are other examples in the book, but you can look at them. But the key thing to know is that it's uninucleated, slow to contract, tires slowly, and is involuntary. So you don't think about it. You don't think about churning your food. I hope not anyway. Cardiac muscle. This is myogenic. Now there are 3 types. There's atrial, which I've put as A, ventricular, which I put as V, and specialised excitatory and conductive muscle fibres, which I put as E plus C. So it's myogenic for me to conduct on its own. You don't need to start it to contract. The brain doesn't need to send a signal to make it contract. It will do it on its own accord. However, the brain can change the rate at which it contracts. So regardless of the brain, the heart will always contract and pump. But if it's too fast, the brain will slow it down. If it's too slow, the brain will speed it up. And that's using the sympathetic and parasympathetic nervous system. The atria and ventricular muscles contract in a similar way to the skeletal muscle, which we'll look at in a bit, but with a longer duration of contraction. The excitatory and conductive fibres contract feebly, but conduct electrical impulses and control the rhythmic heartbeat. They don't need to contract particularly strong because they're just producing an electric wave from the sinoatrial node, and we also have the atrioventricular node. Now, one thing I suggest, I'm not going to go over it now, but if you don't understand about the excitatory and conductive tissues, look at the AS video on transporting animals, because you'll look at how the sinoatrial node and the atrioventricular node work. But it's basically remembering how they conduct. Now, the structure, it is again uninucleated, so each cell has got one nucleus. Now, there are 2 things in here. There are different bands you can see. The red bands, so the thin ones, is what we call it being striated. So it's got those bands. The long, thick black ones, which you'll be able to see quite clearly, are called intercalated discs. Now these are cell membranes, and they fuse in such a way that there are gap junctions with free diffusion of ions and action potentials can occur. Basically it means that you can get action potentials forming very easily, and it can form very quickly between cardiac muscle fibres. Because if you look, it's kind of got a lattice network. If you look at a diagram, you can see this, all connected together through different ways. It means it can pass its wave of excitation quite quickly. And finally, skeletal muscle. This is voluntary, and at the bottom I said multinucleated. It's got more than one nucleus. Now there's a lot of different words you need to know here. And all of them have the root sarco, which means flesh. That's the way to remember it. So we have sarcoplasm, sarcoplasmic reticulum, sarcolemma, and sarcomere. I'm just going to go through what these are and then I'll explain them. Sarcoplasm is basically cytoplasm. A lot of these just happen to be the skeletal muscle version of the word it sounds like. So in this case, the sarcoplasm is muscle cell cytoplasm. There's the sarcoplasmic reticulum, which is the muscle form of endoplasmic reticulum. There's a sarcolemma, and this is the cell surface membrane of a muscle. And then there's the sarcomere, which is the smallest contractile unit of a muscle. The sarcomere we'll look at a lot more in detail on the next page, because on the next page I've got the structure of the skeletal muscle. Now, the sarcoplasm has many mitochondria because it needs to produce ATP, as we'll see later. The sarcoplasmic reticulum, there's a lot of that, and also has, you'll again see why later, and there's a number of myofibrils, MYO fibrils, not microfibrils, myofibrils. These are the contractile elements, and each consists of a chain of smaller contractile units called sarcomeres. Within the myofibrils are two types of protein, and we'll look at this next. Actually, I think the one after next. Yeah. Right. Here is the structure of voluntary or skeletal muscle. It looks quite confusing, but there are two types of filament you need to look at. There's the thin filament, which is the small thin line, and there's the thick filament, which is a long thick line. Pretty obvious. We'll go into what they are later, but I want to get the actual structure. Now, there are four key bits you need to know about. There's a Z line, an I band, an A band, and an H zone. Now, I'm going to do them in order of kind of easiness to understand. The A band is just the length of the thick filament. So as you can see here, the A band fits perfectly with the thick filament. And these thick filaments are in between the thin ones, as you can see. The I band is basically between the end of one thick filament and the start of the next, as you can see. So the A band is the stretch of thick filament. The I band is in between the thick filament. So it'll go A, I, A, I, A, I. Now, as you can see, the thin filament overlaps with the thick filament. This H zone is the bit of the A band, or the thick filament, which isn't overlapped by the thin filament. So you can see there, the H zone, it consists of the thick filament, but any bit of the thick filament that's overlapped by the thin filament is not part of the H zone. And finally, Z lines. Now, the Z line is just almost the beginning and the end of a sarcomere. A sarcomere is just from one Z line to the next, and that's the smallest contractile unit. So one contractile unit is from one Z line to another Z line. It will contain the H zone and the A band and some of an I band, but the I band overlaps them. Now, when the muscle contracts, is you get the sliding filament model, and basically it means that these thin lines will slide over the thick lines more. So as you can see on the second one underneath, you can see that the A band thick filament hasn't changed size. It's exactly the same size, but the actual the small thin filaments have overlapped more. So if you look, the A band is the exact same length, but the H zone is a lot smaller because a lot more of the thick filament is overlapped by the thin filament. Equally, the I band has shrunk, and also the sarcomere has shrunk as well because it's overlapped. Because basically the whole thing is decreasing in size. So when a skeletal muscle contracts, everything shrinks. So the Z zone, the Z lines become close together. So you get a smaller sarcomere, a smaller H zone, and a smaller I band. But the only thing that doesn't change is the A band, because the A band is just a thick filament, and that stays exactly the same size. I hope that makes sense. It's quite confusing, but yeah, I hope that makes sense. So now, what actually is the structure of the thin and thick bands or filaments? The thin filament is made of actin, and there's two types of actin we look at. There is lots of little G-actins which form a strand, so you get a strand of G-actin, and then two strands of G-actin will wrap around themselves like a double helix, like DNA kind of, and this produces F-actin. And basically the difference is G-actin stands for globular actin, so it's like a ball, and loads of these balls join together form a chain. When you have these two chains wrapped around each other, you have F-actin, which is fibrous actin. You then have two other molecules on the thin. You have tropomyosin, which is a rod-shaped protein, and these coil around the F-actin, reinforcing it, and we'll look at more what they do later. And you have troponin, or a troponin complex, which is attached to each tropomyosin molecule. Now, the troponin is a protein which contains 3 polypeptide chains. One binds to tropomyosin, one binds to the G-actin, or F-actin, I mean, same thing basically, Basically, one binds to the actin, and the other will bind to calcium ions. And we'll look why. A lot of this makes a lot more sense when you've learnt the whole of it together. That's why in my next thing I've combined two sections together, but we'll see. The thick filament is just made of myosin, and each myosin has a tail and two heads. And a lot of these are found together in a kind of band with all double heads facing different directions. So basically you've got almost like a cylinder with lots of double heads sticking out around the kind of surface, the curved surface of the cylinder. So now we'll see how these work. Now we're going to look at the power stroke. This is actually how we get a muscle to contract. And there's 5 stages. So thing is to look at. The shaded in rectangle is the actin. The bit blocking that kind of curved bit, which I've done in red, is the tropomyosin, and the little kind of curvy Y on top is the troponin. As you can see, the black circle is a calcium ion. So what happens is, when no calcium ion is bound to the troponin, tropomyosin blocks the active site of actin, meaning that as you can see, the kind of thing that looks like a bit like a sled, which is the myosin molecule, cannot bind to it, because myosin will naturally like to bind with the active site of actin because it forms a very stable complex. But in this case, no calcium means tropomyosin is blocking the active site, so myosin cannot bind to actin. When calcium binds, it causes a change in shape, meaning that tropomyosin is no longer blocking the active site, so the myosin will bind to the active site. Now naturally it has ADP and PI attached to it. What it will do is it will release ADP and PI at stage 3, and this will cause it to kind of, it's quite hard to draw, but it causes it to move backwards. So if you think you've got, I've got one here, it'll be no benefit to you as I'm not filming this, but I'll try and explain it. Imagine you've got like a bit of string, and it's curved in such a way that it is just kind of, well, looks like image 1. It's a little bit bent over, but not very much. Then what happens is you keep one end constant, so the bottom end that isn't curved, hold the curved end and pull it backwards. You kind of get, that's what it's like. It touches the curly end, which I've demonstrated here with a little square on it. And this square, you pull the square back, so the whole molecule moves back. Now, since the myosin molecule is attached to the actin, it causes the whole actin to move backwards. And by doing this, it is causing it to contract. So that's what's actually causing it to contract. So I'm just going to go over step 2 and 3 again, just to clarify. Step 2, calcium binds with troponin. So tropomyosin moves because the shape has changed of the whole molecule, so the active site is free for myosin to bind to. Myosin then moves backwards, causing the actin molecule to kind of move backwards as well. And basically, think of it as the sarcomere that it's been attached to and is moving backwards onto the previous sarcomere, which means that you're reducing that H zone, you're reducing, the sarcomere length. And ADP and PI is released. Now, at stage 4, it's in a very stable shape, as I said. So it requires ADP to break that cross-bridge bond that's formed. So the ADP breaks the cross-bridge bond, and the myosin molecule moves back to where it originally was, so it can now bind to another actin binding site. So I hope that makes sense. If you read on page 236, the power stroke, the 4 stages there, that's what I've drawn here. So basically, calcium binds, shape changes, active site free for myosin to bind, myosin pulls backwards, contracting the muscle, it then ATP bonds to it, causing it to, well actually one thing I missed out, ATP binds to it and is hydrolyzed. Remember, it's releasing energy. It's hydrolyzed to release energy to break the cross-bridge bonds. That's where the ADP plus PI comes from. So ATP binds, hydrolyzes, breaks the bonds, the cross-bridge bonds, and then the myosin molecule moves back to its original shape, but the actin stays where it is. Now, where do we get this ATP from? 2 of them are quite simple, and that's aerobic and anaerobic respiration in the sarcoplasm, and mitochondria as well for aerobic. But also, you can get transfer from creatine phosphate in the muscle cell sarcoplasm. Basically, the phosphate group from creatine phosphate can be transferred to ADP to form ATP very quickly, but it doesn't last for very long. I hope that makes sense. It is pretty confusing. But one other thing, how does this actually get initiated? We said we need calcium ions to get it started, but how do we get the calcium ions? Well, it's very similar to synapse transmission of an action potential. An action potential arrives down the motor neuron and causes almost certainly probably calcium ion channels to open. The calcium ions diffuse into the neuromuscular neuron, binds to vesicles containing acetylcholine. They fuse with the neuromuscular membrane, secreting ACH or acetylcholine into the cleft. It binds to receptors on the neuromuscular membrane, which will cause something called the T systems to be depolarized. So it travels down these tubules, which are the T system, depolarizing it. Now this depolarization goes to the sarcoplasmic reticulum, which then releases calcium ions, which then will bind to the muscles. So up until, you can see the numbers here, 1 and 2. 1 and 2 is very similar to just synaptic transmission. Basically, calcium ions diffuse in binder vesicles containing ACH, which fuse to the neuromuscular junction instead of synapse, say neuromuscular membrane, causing acetylcholine to be released into the cleft, bind to receptors, complementary receptors on the muscular membrane. And then at stage 3, basically the T system depolarizes, causing the sarcoplasmic reticulum to release calcium ions and then bind to a muscle. And to make sure this doesn't happen continuously, just like in a synapse, acetylcholinesterase in the gap breaks down acetylcholine, so the contraction doesn't occur forever. I hope that makes sense. Basically, if you understand synaptic transmission, it's very similar. Just replace it synaptic with neuromuscular, and just know the T system goes to sarcoplasmic reticulum, which releases calcium ions. One thing to know about is the differences between neuromuscular junction and synapses. So I'm going to name a few now. There are a few more as well, I'm sure. The neurotransmitter in a neuromuscular junction is always ACH, but in a synapse it can be various. It can be ACH or noradrenaline. The receptor in a neuromuscular junction is called a nicotinic cholinergic. That's it. Nicotinic cholinergic. I'll say that one more time to actually say it right. A nicotinic cholinergic receptor. In a synapse it's various. In a neuromuscular junction there are lots of receptors. In a synapse there are fewer. In the membrane, the clefts contain ACHE. So they have the neuromuscular junction membrane has lots of clefts in it. In a synapse it's very smooth. And the effect of neurotransmitter will always cause a muscle to contract, but may or may not start an action potential in a synapse, depending on the amount. Now, the other thing to mention is the motor unit. Now, the motor unit is quite simple to understand. It's basically that some muscles need to contract stronger than others. So you have lots of these different receptors, or motor units, and the stronger the contraction is needed, the more signals are sent to these motor units. So basically, more motor units contract. So you get more, the muscle will contract stronger. And finally, the fight or flight response. This is basically the response to stress. I'm firstly going to talk about the physiological changes. The pupils will dilate, and I want you to think, I'm not going to say this until after I've gone through all the responses, but I want you to think, what type of muscle is controlling this? Is it going to be cardiac, involuntary, or voluntary? Voluntary. So pupils dilate, heart rate and blood pressure increase, arterioles to the digestive system and skin are constricted, while those to the muscles and liver are dilated. Blood glucose concentration increases, metabolic rate increases, rector pili muscles in the string contract, ventilation rate and depth increases, endorphins are released in the brain, and sweat production increases. Now, to name some of them: pupils dilating, arterial to the digestive system and skin constricting. Those are obviously involuntary, you don't control them. Heart rate and blood pressure is cardiac. So things like that, because they could ask a question on that. Now, generally, you won't get many physiological changes which are voluntary, because you can't really cause them to happen. So they're going to be involuntary or cardiac. So what actually happens? You have a stimulus. A stimulus will occur, and it will be registered in the cerebrum, because you know we go to those association areas to see what should we actually do. Then it will send a message to the hypothalamus. The hypothalamus has two effects. It goes to the sympathetic nervous system, because it's a stress response, not a relaxed response, a stress response. And the sympathetic nervous system will do two things. It will activate glands and smooth muscle. So as we said, glands, it will release, blood glucose concentration hormones. So you want to increase blood glucose concentration. So that's going to be glucagons released, metabolic rate increases, all these hormones. But also it's going to go to the muscles, like we said, in the digestive system, because you don't want those, contracting as much, because you don't want to waste energy on them. But also the sympathetic nervous system goes to the adrenal medulla, which will secrete noradrenaline and just normal adrenaline, which will then enter the blood and cause a response. The hypothalamus also, though, sends messages to the anterior pituitary gland, and this secretes the hormone ACTH, which is adrenocorticotropic hormone. Sorry if I said that wrong, but yeah. This hormone stimulates the release of a number of different corticosteroid hormones from the adrenal cortex, and some of these hormones help the body to resist stressors, and the stressor is a stimulus that causes the stress response. It causes wear and tear in the body's physical and mental resources, so you don't want it happening too much. You want it happening a little bit to get the actual response started, but not too much. So this anterior pituitary gland releases ACTH, and that goes into the blood, causing a response. So basically, stimulus, cerebrum, hypothalamus, which affects the sympathetic nervous system and the anterior pituitary gland, which will cause either some hormones to be released like ACTH, noradrenaline and adrenaline, or perhaps ones that are found in glands, and also stops smooth muscles when they don't need to be contracting, and smooth muscles to contract when they are needed. So in conclusion, you need to know about the brain, the organisation of the nervous system, 3 types of muscles, smooth, skeletal and cardiac, and the fight or flight response. Now this is a confusing module, I think, or definitely a very confusing topic, and I hope I made sense of that. So as usual, email me if you don't understand, or leave comments or anything if you didn't understand something you wanted differently explained. So thank you for watching, and good luck on the upcoming exams, everyone. Thank you and goodbye.